The initial code that turned into CompuCell was something that I wrote when I was a graduate student back in the 1980s. And it was turned into, it was originally written for material science applications. It was turned into code for simulating tissues back in the early 90s. When I lived in Japan, I was working as a postdoc in Sendai in Tohoku University. And we wrote a couple of papers on modeling tissues using these particular computational methods. In those days, it was all old Fortran code. And then I continued to develop that work when I became faculty member at the University of Notre Dame, as it happened. And then we had the opportunity more than 20 years ago to take that Fortran code, which was very old and not very shareable, and turn it into something that would be an open source public resource. And that led to the creation of what was called CompuCell. And that was written in C++. And their legacy code from that, that's in the code that today. And then when I moved to Indiana University 20 years ago, Maciek Swat became the lead developer for CompuCell, and he refactored the code and created what's now CompuCell 3D, which is Python scripted. And that's descendants of that code are still here today. The original algorithms, the core algorithms, many of them still are descended from that Fortran code that was written back in 1988 or 89. But I don't know whether there are any lines, explicitly lines of code that survive, but some of it was directly translated from Fortran to C++. So probably there's still some original lines that are there. So it's an old project. Well, I'm glad to hear we have a biologist in the room. We'll try to make things interesting for you. Today, this is going to be a very, the point of today really is just to get people's feet wet, get people started, potentially answer questions if there are any questions, and make sure that people have the support and resources that they need so that when it comes time for the workshop proper, everybody can enjoy it and be successful with it. Canonically, it seems that 3 past the hour is the start of the hour. So I'll give 1 more minute and then we'll get started. Someone says, you've downloaded it, but you don't know how to run it. If you knew how to run it by yourself, I guess we would need the course. So I hope at least by the end of the course, you know how to use it. At least a little. It's a very flexible package, and it continues to grow. And so I have to say the parts of it that I've never used. Probably the only people who know all of CompuCell are TJ and Maciek, who were the 2 developers. And people are always surprising us. They're always using it in new ways that we didn't think about. It's one of the pleasures of working with it. Okay. All right. Well, people may join us a little bit later, but I'm going to get started. So I want to welcome you all to our 3rd of the day orientation for the CompuCell multiscale virtual tissue modeling workshop and hackathon that's going to start the first week of August. I appreciate you coming tonight to try a little bit together and talk about it. I should say that this meeting and the workshop itself are live streamed on YouTube, and the recordings will be made available publicly. So please remember that. If there's a problem, let us know about it. One thing that's very helpful is to have CompuCell running on either our desktop or NanoHub. We're going to do almost all of our teaching through the NanoHub deployment. The NanoHub deployment has the advantage that it's a uniform interface. There are some minor glitches that we'll run into, perhaps, that you'll see perhaps tonight. They happened earlier today, but it does mean that people don't have to do an install. So that sometimes makes life a little easier. I'm going to put again in the chat the links for the Zoom sessions for the main meeting and hackathon. And here's begin with, I'll just give you an outline of the course. I'm going to give you a very quick welcome, and then we'll talk a little bit about the logistics of the course. I'll do a very quick introduction to CompuCell and how it works, and we'll actually build a tiny simulation and run it together. And then we'll talk a little bit about what you might want to do to prep for the main course. We have more than 150 people who've registered for this course. That's a lot. I'm sure not everyone will show up, but it's a much bigger course than we're used to, and we'll do our best to support each one of you with the teaching staff that we have. And so we do appreciate your patience with that. This is a bit of an experiment. So as a welcome, I want to just give you a bit of an idea about what we think of when we think about the kinds of things CompuCell can do. CompuCell is fundamentally a multicellular tissue modeling package. And so it was developed originally to try to understand developmental biology processes at the cell level, how do cells organize to form tissues. And over the years, it's been used to study developmental biology. It's been used to study homeostasis, how tissues maintain themselves. It's been used to study developmental diseases, in particular cancer. Most recently, people have used it quite a bit to model infection, viral spread, and immune response. The Environmental Protection Agency has used it to try to model toxicological problems, and it's been used to do models of tissue engineering as well. All of these are situations in which you want to understand how individual cells interact with their environment to provide the functions or dysfunctions that you're interested in. Biology exists across many, many different length scales, of course, from nanometers up to meters, depending on the organism. The simulations that we're going to be working on tend to focus on 3 scales, although they can be extended both up and down within the package. The molecular networks, gene regulatory networks, signaling networks, and metabolic networks that control cell behaviors. Those cell behaviors and how cells move and interact and exert forces and do things. And then the environment that the cells find themselves in, the extracellular matrix, the medium, the chemical fields around the cells. And this is a simulation of, it's blocked today. Don't know why the simulation doesn't want to run today. This is a simulation, which you're not going to see, of some segmentation of the early embryo. And it's designed to be, it's supposed to run. There's always a glitch, it never fails. It's supposed to run because it actually has a subcellular chemical oscillator running inside of each cell. Those chemical oscillators interact with morphogens in the extracellular environment, and that gives rise to cell differentiation and to reorganization of the tissue. And you'll see this simulation in the main class, and I promise it'll work then. Somebody says try again. It's giving me a message saying it's unable to play the media. So it's a permission problem for the file, I'm afraid. Course logistics. For the Python Bootcamp and Hackathon, let me give you, I will put in the chat, there is a Slack for discussions. There are Zoom links for the Python Bootcamp, the main course, and the Hackathon, which I will put in here. Those will also be available in the emails we send and the websites. If you like, you can check to see whether you can register successfully for the various modules of the course. If you want to participate in the hackathon, we definitely encourage you to do that. We want to help people team up. And so in order to do that, we have a document, just a Google Doc, where you can type briefly about your idea. We're going to ask people to also put their idea into the Slack to try to recruit teammates. And we'll ask you also to upload a single slide, a single PowerPoint slide for a lightning talk to pitch your idea on the second day of the main workshop. We'll do our best to make sure that we build teams that work and can be successful in producing a result from the hackathon. Logistically, the microphones will typically be off during the sessions, and screen sharing will be off. We'll have breakout rooms where people can have open conversations. We'll also have opportunities for open discussion, but with a very large group. Sometimes it's easier to do it this way. Please remember to use the various things that Zoom provides. Yes, no, go slower, go faster, raise your hand, and these kinds of things. We'll do our best to provide exercises in Google Docs so that they're available offline, also demonstration code offline. And I will be continually and perhaps tediously polling people when we're doing exercises, asking you whether you're okay getting through the exercise, whether you need help or whether you need more time. And we'll try that out today just to make sure that it works. There'll be videos and PowerPoints available for all of the presentations. And as I mentioned, all of the presentations are live streamed and recorded. The list of instructors may vary a little bit, but the current list of instructors are myself, James Glazier from Indiana University, Jim Sluka, also from Indiana University, who is on the call tonight, helping us out. TJ Sego, who's the current lead developer of CompuCell, also from IU. Professor Giulio Belmonte from North Carolina State. Professor Gilberto Tomas, who joins us from Brazil, and who's been involved in the CompuCell project for more than 20 years. And then we are also going to have two graduate students who are expert CompuCell users, Giuliano and Joshua from IUB. And we hope also some contributions from Professor Bobby Matamachi of University of Michigan, who's an expert in science education and also a great CompuCell user and has contributed a lot to the design of the course over the years. You've got our emails, and you're always welcome to contact us for help, either directly by email or through the Slack. The course itself will have the same rhythm for 6 consecutive days. We will have a half an hour before the official start of the meeting when we can have informal discussion, troubleshooting, questions, and so on with the instructors. Then there'll be 4 1.5-hour modules. Each module will consist of half an hour to 45 minutes of presentation and 45 minutes to an hour of exercises. We are trying to avoid having walls of text and have people engaged as much as possible. Maybe the very first module will be mostly lecture, but we will try to avoid giving a lot of lecture in a block. We'll have 15-minute breaks between the first 2 and the third and fourth modules, and then a slightly longer, quote, lunch break in the middle. After the regular session, we'll also have a half an hour for questions and discussion and troubleshooting. So we'll begin and end with those. I hope that rhythm will work for people. It's actually a little bit less course time than we had in previous years. But people found that Zoom for 10 hours a day was pretty exhausting. And so we're going to try to be more efficient in our presentations. There's a tentative course outline. The details may change a little bit. We have day 0, which is Sunday, August 1st, which will be a Python tutorial refresher. If you're not an experienced programmer or Python user, we strongly recommend that you take that. We will cover all of the Python you need in the class proper, but the class moves pretty fast. And so if you haven't used Python before, that's a good opportunity to bone up on your Python familiarity. Day 1, we'll have an overview of some example models. We'll begin working with Compusel, learning how to use Compusel to make cells and have them do things. Day 2 will be focused on making cells move, a little bit of hackathon organization, and then we'll talk about building the coupling between cells and those extracellular chemical fields. And that will lead to discussions of cell growth and division and building a simple model of an avascular tumor spheroid, which is a good base to build all sorts of different kinds of models. Towards the end of the day, we're going to deal with cells that have internal structure. The simplest cells we'll model will be basically blobs of material, which do things. We'll have Gilberto Tomas from Brazil talking about how to model nuclear compartments and cytoskeletal activity in cells and how you can do cell migration simulations with that. Day 4, we'll talk about static or persistent junctional structures, what are called links, that you can use to represent things like focal adhesion plaques, filopodia, and all sorts of complex force generators. And we'll then talk a little bit about doing things like parameter identification, parameter scanning. And the last day of the class will be devoted to understanding how to do subcellular control, how to represent subcellular networks, and the way that they control cell behaviors. The hackathon will be the 2 days of the weekend immediately after the main course, Saturday and Sunday, the 7th and 8th of August. We will try to have teams self-organize. We hope that they'll coalesce. Over the years, we've had a lot of hackathon teams that went on to publish the research they started during the hackathon. We hope that will happen again this year. We'll begin each day with a brief check-in and have a noon check-in as well, just to make sure things are going okay. We'll have instructors available to circulate and help people out. At the end of the first day, we'll have very brief reports from each team. At the end of the second day, we hope people will have little mini presentations of their results, and that we can then go forward after that and continue to help you develop your ideas into something that you can publish. The hackathon teaming, as I mentioned, you can start that right now by posting your ideas to the Slack channel. We have that shared Google Doc. We encourage you to post your ideas there. And then also, please send us a single PowerPoint slide with your idea by Monday, the end of Monday, the first day of the meeting, which is August 2nd, so we can put that together and have people give 30-second, 60-second presentations of your idea to recruit a team. We have to do it that way because if we have 75 or 80 presentations and we have to switch and have each person screen share, it will take the whole day. So we have to have the slides in advance. Coming to resources, there are a number of places you can go for help, online help. The Compusel website has manuals online. The class files will be distributed through the Slack channel. There's a course Google Drive that I hope people have found where we'll have everything posted. And we'll also try to mirror that on the Compusel website, although putting it up in the HTML for the website sometimes takes a little bit longer than putting it in the Google Drive. So that's why we have multiple sites. For subcellular modeling, will be using primarily the Tellurium modeling language. There's a very good online manual and tutorial that's available, and we encourage you to look at that. And then there are also some quick reference guides that we will post. And let me put the links for these into the chat. So here are the Hackathon and Google Drive. Here are the Hackathon links, and here are the links for the material related to the manuals. So I hope everybody can use those, okay? And I'll put the Slack in again. The Slack is there as well. So I hope that those are helpful resources for people. The Slack should be monitored, and but it really is only useful if you use it. So you have to decide whether the Slack is a channel of communication that you like, but we will do our best to make that a helpful session. I want to call your attention to the quick references. There are 3 of them, and I encourage you strongly to print them out. If you have them on a piece of paper on your desk, you don't have to switch back and forth between screens or tabs on your computer, and it makes life much easier. I always keep them printed out on my desk. There is a beginner's Python cheat sheet, which covers the basics of the Python language in a very compact way. Everything you're going to need to do about Python almost is there, and it's a very well-written document. For the subcellular modeling, there's a Tellurium cheat sheet. Tellurium is actually a very simple language to use. And if you just look at the first page of that cheat sheet, you'll actually know pretty well how to model subcellular networks. And finally, there is a quick reference guide to CompuCell, which describes a lot of the important functions and features of CompuCell with some basic examples. And I think that that's also useful to have printed out as well as available online. If you want to dig a little bit deeper, there is an article that we've posted in the Google Drive on how somebody asked how CompuCell got started. It's an article on the history of CompuCell and specifically on the methodology that CompuCell is built around. If you have a little bit of physics and mathematics background, I hope it'll be interesting to you. If that's not your thing, don't worry about it. You certainly don't need to know the numerical and physics details of how CompuCell works for it to be useful. But if you're interested, it's there. If you haven't tried building mathematical models of biological systems, we just saw a paper from a group of students in Canada called 10 Simple Rules for Tackling Your First Mathematical Models. It's not really about multicellular modeling. It's more about classical biological modeling, but it seems to us to be a pretty good introduction. And I recommend it if you're unfamiliar with the concept about how to think about building models. So we posted that also in the student materials. And there's also a document that I wrote talking more specifically about building multicellular models that you may find useful. So those are some resources. Now I'd like to talk a little bit about CompuCell itself. CompuCell is a software tool. It's a software environment. It has 2 pieces that you're going to be interacting with. The first one is called Twidit++, and Twidit++ is an editor like Microsoft Word. It's an editor specifically designed to build multicellular models. And you'll find that Twidit++ has a lot of features that are designed to make simulation coding a lot easier. So if you're not very familiar with Python, you're not familiar with XML, Twidit++ has a lot of built-in features that help you learn how to do it. Most of the things that you do typically in CompuCell are templated. So you say, I want to do something to every cell of a particular type. The code that you need to do that is there, and you don't have to figure out or remember how to do that yourself. So there are a lot of intrinsic features in CompuCell that make it easier to build complex simulations. CC3D Player is what it sounds like. It runs and displays the output of simulations that you've specified using the model editor, model Twidit. And so Player and Twidit work together essentially to allow you to specify and then execute models. And we'll work with both of them a little bit today. Installing and running CompuCell, as I mentioned, we're going to be teaching almost entirely out of the web-based deployment on NanoHub. Over the years, we found that sometimes people have difficulty installing CompuCell on the desktop machines. We will definitely help you if that's the case, provided you're not on a strange Linux box. We do have some deployments for Linux, but everyone's Linux is different, and we can't promise that that will work. Occasionally, we've also run into people who have Macs that have odd configurations that are difficult. The most recent versions of both Windows and Mac operating systems have made it more difficult to run third-party code. And so if you try to do a local installation, you may find that you have to override protections, temporarily turn off virus protection features, or you may have to run the installer as an administrator. If you're going to have that issue, then if somebody's saying that there's a Slack link problem. Jim, could you repost the Slack invite? Because I've had at least 50 people today successfully join the Slack, so I'm not quite sure what the issue is with the Slack invite. Do I have a stale version of it? Okay, somebody says the one from the slides works. Let me, Jim, can you check that out and make sure that I'm sharing?
I just did, and it works for me. So I don't quite know how to fix it when it already works for me. So I don't know if there's some strange copy paste error or, but someone said they just had one working. We'll ask them to post it to the Slack.
Okay, whoever was the last person who said that they were able to get into the Slack, could they?
Yeah, I got you.
Okay, thank you. I'm not quite sure what's going on there because I think people are using the same link. And I certainly don't want people to have a problem. If you do have a problem, email us and we'll check it out. And we can send you an invite from the Slack if we have to. It probably isn't. So why don't people try that invite and let us know if that works. Did that work for people? Yep. Okay. I'm sorry about that. Occasionally these things happen and I can't, I'll do my best to troubleshoot, but I'm not a Slack jock, so I'm not. No, there's absolutely no reason to build it from source, period. Unless you're masochistic, you don't want to build it from source, I promise. The only reason to do that would be if you're on a weird Linux installation, which is non-standard, you might have to. But the Windows installs work very well, and the Mac installs work very well. That's one of the big advantages of CompuCell is it has a one-button install. Now, if you want to develop core CompuCell code and become a software developer for us, that would be wonderful. So we'd be happy to work with you on that. Okay. So let me just show you, for those of you who don't want to try to compile CompuCell, let me show you to begin with how to run CompuCell on NanoHub. because that's the easiest thing to do. And I'd like to ask people to follow along on these exercises. All right. And so I'm going to show you a little bit about how, just introduce you to using NanoHub tools. And as I mentioned, NanoHub has some real advantages. You don't have to install any software on your computer, and you'll have a uniform interface. However, it also has some minor glitches. And so we'll just have, there's some trade-offs. If you want to go to the CompuCell NanoHub page, the NanoHub link is here, and I will put that in the chat for you. And so I'd like everybody to try that. If you have already got a NanoHub account, that link will take you directly to the CompuCell site on NanoHub. If not, you will get a page that looks like this, and you will have to create an account. And creating an account is not difficult. You don't have to be in the United States to create an account. You don't have to be an academic to create an account. However, if you are an academic in the United States, when you hit login, you will be given a variety of sign-in methods. If you're in an American institution, you can click on sign in with an affiliated institution and then type in the name of your university. And it should recognize your credentials for the university, and that should work. If you're not at an American institution or you don't want to use university authentication, you can sign up with Google or with LinkedIn, and there's some other alternative ways of signing in as well. And so I'd like people now to take a few minutes and make sure that they have a NanoHub account working. And I'll do my first poll, which will be asking you if you can get into NanoHub. And I would say with these polls, there's an option called need more time, but I wouldn't use that one until we've had a few minutes, because once you've clicked need more time, you can't unclick it. And so in order for me to make sure that everybody's done, I have to relaunch the poll, which is fine. If you need help, you can ask for it there. And occasionally people are just listening along, and it's then if you click not doing the exercise, we don't wait for you to finish. If somebody has a problem, please speak up. You're always welcome to ask for help. And we'll always have some breakout rooms and people available to help out with technical issues, which are not something we can, if there's something we can't deal with in the main section. So are people able to get into NanoHub all right? It sometimes registering takes a minute or two to do. If you're finding you're not able to register, let us know and we'll do our best to try to help you out. So once you're in NanoHub, you will see, sometimes, depends on how you've signed in, you'll see different things. NanoHub hides one of the things that you need. If you go to the top right-hand corner and click on it, you want to click on Dashboard. And Dashboard will pull up a set of navigation tools. The interface is not the most elegant, but it works. And the one you want to look at, and I'm going to probably resize the screen a little bit to give a little more room to the NanoHub box here. What you want to look at here is, again, that link that I sent you for CompuCell will work. You can also search, if you go to all tools, you can type in CompuCell. And then you would want to do CompuCell 3D V4. And you want to make sure that you're actually running CompuCell 3D version 4, because there are older versions of CompuCell lurking on NanoHub. They never get rid of things. And you want to run the up-to-date one. And you want to bookmark that. Bookmarking that is hitting the little heart button. That'll bookmark it to your page. And then when you've got that going, you would click the Launch CompuCell button here. And you should see a timer and then a blue square. And that shows that CompuCell is launching. And if we're lucky, CompuCell will start running. And if NanoHub is having a problem, we may find out that CompuCell doesn't run, but it should be running. It normally starts pretty quickly, so I'm a little surprised that it's taking its time. There it goes. So it should launch the blue square, and then you will see a box inside of it. And that box here is CompuCell Player. So do people have that working? If you were using, I'll show you how to launch the desktop app in just a second. Does everybody have CompuCell up and running? If anybody's having a problem, now's the time to speak up. Okay, somebody said they need help.
I'm trying to find in my tools. It's CompuCell 3D V4.
Yes, with a space.
Okay. Okay.
You find it?
Just a minute.
You can also go from outside and hit this link. Where was the link? Here, I'll put this link again. If you click on the link from the external link, once you've logged into NanoHub, I put it in the chat again. That should take you directly to that page.
Okay. It's fine.
Great. Got it? Great.
Yes. Thank you.
Okay, because I know there's nothing more frustrating than to have whoever's in front of the room continue on when you're stuck. So I don't want to have that happen. That's good. Okay,
Yeah.
good. Great. Okay. All right. If you want to install CompuCell as a desktop app, and again, I'm not particularly encouraging you to do that right at the moment, although you certainly can. You will go to the CompuCell webpage. You will click on binaries here. And then you will decide if you're Windows, Mac, or Linux. I happen to be Windows. I click on Windows, and I'll go to download Windows here, and that will take me to SourceForge. And SourceForge should recognize what kind of computer you have, and you should be able then to download the latest version of CompuCell. As I mentioned, when you do that download, you may find these days that Windows gives you a warning about uncertified software. It may tell you that you have to do the install as an administrator. It may tell you have to put the installation in a particular place. And I had an issue the other day with the newest version of CompuCell where I had to do the download twice. The first time I did the download, the installer didn't run. It threw a warning. I downloaded it again as a clean install, and it worked fine. So if you have problems with the desktop install, we're not going to have time to debug those now. But between now and the time that the course starts, please do contact us we'll do our best to get you running. It usually really is a one-button install. Especially on Windows, it works. Now, once you have it installed on your desktop, you have to find it. My recommendation is to search in your Finder, either on Windows or on a Mac. Search for the editor, Twidit++. And the reason for that is that that's a unique name, whereas CompuCell, there are a lot of components that are named CompuCell, so it makes it a little harder to search it. Twidit++ and open it. And now we're going to spend a few minutes actually writing a CompuCell model. And it's going to be a lightning version of that, so we're not going to go into a lot of detail. But I want you to have that experience, and then when you come back, it won't be unfamiliar. So we're going to use Twidit++, and it has a tool called Wizard that helps you rapidly build the basic configurations of a simulation. Typically, you're going to have to edit things after Wizard is done, although today we won't. We're actually going to use the default settings from Wizard. We're going to do it in NanoHub, although the same thing will work perfectly well. Yeah, we can't help with Linux now. We'll have to do that offline. So let's just stick with the NanoHub for today, okay? We'll be happy to hook you up with a Linux expert offline. Jim can certainly help with that. And I hope we can get it running for you. It should be okay. For Ubuntu, it should be all right. So let's just launch Twidit in NanoHub. We have player open here. We want to switch to Twidit. To switch to Twidit, you have a couple of options. If I go under File, I can do Start Twidit, like that. I can also hit this icon that is supposed to be a piece of paper and a pencil that suggests an editor, which will also start Twidit. So let's do that. And we'll see a new screen here, which is the model editor. One thing that you may find in NanoHub is that the screen redraw in NanoHub doesn't always work. That's not a service of CompuCell, it's a service provided by NanoHub. And so it's not something we have control over. And so if you see that your screen hasn't redrawn properly, hover over the bottom right-hand corner of the screen. You'll get a four-headed arrow on that. If you drag the corner just a little bit, sometimes it takes a second to have it pick up properly, it will redraw. And so if you ever get a distorted screen or a misdrawn screen, try joggling that corner and forcing a redraw. So that's what I'm saying about sometimes having the minor annoyances in the NanoHub deployment. Okay. To start building a simulation, we have to tell it that we want a new CompuCell project. And to do that, we go to CC3D project here. And I want everybody to follow along with me and select the first item, which is new CC3D project. And you should pop up a window that looks like that says CompuCell Simulation Wizard. Let's make sure that people are able to get that to work. Okay, it looks like everybody's okay with that. And so now you have to give your simulation a name, and we can call it just My Simulation. The one thing you want to be careful about is you don't want to have any special characters and don't put spaces in. On a Windows box, you can have spaces in the file name. Linux dislikes spaces in file names. So don't do that. You also have to check for your simulation directory. It should automatically point to your home directory in NanoHub, but to check, you hit Browse, look at Computer. You don't want to point at Computer, find your name, and select Choose. So you have pointing to your home simulation directory here on NanoHub. If you're doing it on a desktop, you can point to any writable directory that you have access to and the name of your simulation, and then hit Next. Okay. The first panel, General Simulation Properties, specifies things like how long the simulation will run for, whether it's two-dimensional or three-dimensional, what the initial conditions will be, and so on. For the moment, we'll accept all of the defaults, since we don't have time to do a lot of programming tonight. And so we'll just hit Next again. Okay, everybody got through that all right? So the next window that you'll find is to define what kinds of cells you have. This could be epithelial cells, cancer cells, endothelial cells, vascular cells, and so on. For our purposes, we're just going to create two kinds of cells. We could call them just cell 1, hit Add to add it to the net list, and cell 2. You have to tell CompuCell nothing exists in a CompuCell simulation unless you ask for it. And so if you don't tell it that you're going to be using two cell types, it doesn't know that. So you have to tell it. So type cell number, cell 1, add, cell 2, add, and then hit Next. Sorry, I got the wrong slide here. So again, type cell 1, add, cell 2, add, and then hit Next. Everybody got that all right? Somebody says they need help. Okay, what can we do for you? What's the freeze option about? The freeze option says that the cells don't move. Sometimes you want to build a wall, where the cells are not really representing biological cells, but might be representing extracellular matrix or the test tube or something else that doesn't move. And so freeze is an option that says that the cells are not dynamic, that they're static. Are there any other questions on this? That's a good question. Okay. Did somebody say they needed help, or was that the question about freezing cells? So the next screen, which we're going to skip, says chemical fields. If you wanted to specify oxygen, glucose, and so on, we do it here. In our little simulation, we're not going to have any extracellular chemical fields, so we just hit Next and skip it. The next screen is the one that's most important because it specifies what the basic properties of the cells are going to be. There are many additional things that the cells can do that aren't listed in the screen, but the ones that are most commonly used are here. And the first thing we have to do is say that our cells are going to stick to each other. So we click this contact box, and that says that there's a contact energy or adhesion energy between the cells. The second thing we're going to do is say our cells grow. So we click the growth box. We're going to say that the cells divide. So we click the mitosis box. And it may seem that once we have cells growing, they have a volume, but again, nothing exists unless we tell it. So we click volume box here, volume local flex box there. And I want to make sure everybody can do that on that screen. And once we've got that working, we'll hit Next. Is everybody okay with that? That was interesting. On the poll, when I clicked on the poll to restart the poll, I got a Zoom error. I think that's the first time I've ever seen Zoom throw a, it looked like a core dump type error. Interesting. Okay, great. So now we hit next. It says configuration complete, and we hit finish. And what you should see is this, and your simulation is here, hidden behind this navigation bar. And so we are going to double click on Mysimulation. And we will see the actual code that was generated by Wizard here. We're not going to work with that code at the moment. In a few minutes, we'll come back and very quickly explain what that code does. At the moment, we're going to run the simulation. And to run the simulation, first I'm going to say about saving. We don't need to save it now. It's saved temporarily. But if I wanted to save it under a particular name, I can use this Save CC3D Project button here. I can use this double floppy disk icon here, or I can go under CC3D Project and do Save CC3D Project or Save CC3D Project as, if I want to rename it. So I have a variety of options for saving, which we'll be using a lot, especially for version. To run the simulation, if you're on a desktop, this CC3D icon is probably blue, and you can click that to run your simulation. Typically on NanoHub, we will hover over Mysimulation here at the top. We will right-click and hit Open in Player. And once we've done that, we have to switch in the tabs and Twidit, from Twidit to Player. So you click the other tab here, and we can resize this to make it more visible. And you should see the simulation running. So I want everybody to try that. Again, hover over the name of the simulation at the top of the navigation pane. You click Open in Player, and then you want to resize the player window so that you can see it appropriately. And everybody should be able to get that. Make sure that everybody has that. Okay, congratulations. You just created and ran a CompuCell simulation. Not so bad. Are the assigned colors random? Good question. CompuCell will remember the last color assignment you did. And so your colors may vary, and we'll show you how to change colors later. If you want to figure that one out on your own, this icon here that's called configuration that shows a wrench and a screwdriver, not the most appropriate things for computing, will allow you to change your color scheme and a variety of other things. Tonight, we're not going to have time to do that, but that's something you're welcome to explore on your own and ask questions about during the next 2 weeks. Okay, let me give you a few basics about how to use CompuCell in NanoHub. You can resize your windows the usual way by dragging corners, by using maximize and minimize. Within the CompuCell window, you can change the size of the region devoted to this property editor and to the console. A lot of times we don't care very much about what's coming out in the console or the property editor, so you might shrink those to have more displayable area. There's also a convenient button hidden here called Tile that will relay out your console, your display. In this case, we only have a single image, so it's not going to do very much. But if we had many windows, that tile function can be useful. One limitation is the tile function is not available on Macs. Their windowing system doesn't allow that functionality. It's one advantage we have on Windows or on the NanoHub deployment. Again, if there's an issue with redraw, joggle the corner of the simulation to get it force or redraw. The basics of using player, there are these 4 buttons here. Run, which will run the simulation. Stop, which will stop the simulation and reset it to the beginning. Step, pause, which will temporarily suspend execution and allow you, and step, which will take you forward 1 time step in the simulation. So if you've got this running now, and it looks something like this, if you hit stop, that should restart the simulation. Now hit step. This is the redraw problem I was talking about. And you should get a new version of the simulation with the initial condition visible. I hope that worked for people. So you should see something like that. And now I can hit run, and you'll see the cells grow and divide. Did that work for everybody? So this is a simple simulation of cell growth and division. I want to take just a minute to walk you through what CompuCell code looks like, model specification code looks like. CompuCell has 2 components, a CC3D-ML component, which specifies the static aspects of the model, and the Python specification of the dynamic aspects of the model. To look at the simulation specification, we go back to TwidIt. We do that by clicking on that button at the bottom that takes us to the other tab. And within TwidIt, we have a navigation pane. And the 2 things that we're going to be looking at will be this one, mysimulation.xml and mysimulationsteppables.py. And we'll look at mysimulation XML first. To look at that, we can get there in 2 ways. We can double click on mysimulation.xml, or we can just single click on the tab at the top. We can scroll through the simulation using the scroll bar at the right. And now I'm going to give you really a lightning overview, and I realize it's not going to cover things very much detail, but at least you'll have seen what the code looks like, so it won't be unfamiliar for you playing with it yourselves or when we come back to it in 2 weeks. At the very top of a CompuCell simulation, it'll say CompuCell 3 revision number, and it'll give you the version number of the code you're running. The first block will be something called metadata, which is mainly about parallelism in the simulation. CompuCell intrinsically is parallel, and if you have multiple processors, it can take advantage of that. We're not going to play with that right at the moment. The second block, called pods, defines the size of the simulation, because it's a lattice-based simulation, how long the simulation runs for, and things like that. The next block is one which we did do something to. It says cell type. It's called a plugin, and you'll see plugins a lot. Plugins are intrinsic C++ functions that run the core functionality of CompuCell. And here we're defining the cell types. We're saying we have a cell type 1 called cell type 1 called cell type cell 1, and a cell type 2 called cell 2. The next thing we see is that the volume of the cells will be specified. That's all we're doing there. And then we have a statement that the cells have a contact energy with each other and the specification of each pairwise possible contact energy. There are other ways to specify adhesion, which we'll go over in the class at the moment. The last thing we have in the XML is the creator of the initial condition. This is not called a plugin, it's called a steppable. Steppables are called less frequently. In this case, it's only called one at the beginning of the simulation. Uniform initializer creates a rectangular box filled with cells. I can specify the coordinates of the minimum and maximum corners of the box. I can say whether the cells are all packed up against each other or have gaps between them, how big the cells are, and what kinds of cells I want. And that draws that square initial condition that we saw when we ran the simulation. That's the end of the XML. We switch to the Python by clicking here at the top, mysimulationstepables.py, or double clicking here in the navigation pane. I'm not going to go into the Python details, but if you're interested, we can certainly come back and we'll be doing a lot of this later on. And you can certainly read up on it on your own if you like. The main things that we're going to see here is that we have a start function that's run once at the beginning of the simulation, and we're going to use that to set up the initial conditions of the simulation. And I want to call your attention to something that you'll see a lot in CompuCell, which is this command for cell in self.celllist. Cells are objects. For cell in self.celllist says, do something to every cell in the simulation. And what we're going to do is attach to each cell a target volume and something called lambda volume, which is an inverse compressibility of the cell. And you might say, why don't we give the cells a volume instead of a target volume? And the answer is, if you think about it, we can't specify the volume of a cell. All we can do is specify, for example, the mass of the cell or the target volume of the cell. If you think about it this way, suppose we have a balloon, and the balloon is sitting on our table and it has some size. Now we take that same balloon and we take 100 of them and we try to squeeze them into a box. If we squeeze them, the balloons will be smaller than they would be if they were sitting on the table. Think of the target volume as the volume the cell would have if it's not being squeezed, and its actual volume at any time is the result of all the forces that are acting on it. And lambda volume tells you how the cell reacts to being squeezed. And so that's a concept. This idea of target volumes and constraints is something that may be a little bit alien to people, and it's something that we'll cover in detail during the class. The next thing we're going to see is a step function that's called once per time step in the simulation. Again, we loop over all cells, and here we add to the target volume 1. So this is growing the target volume of each cell. And you could play with that target volume in the simulation if you like it, and the rate of growth. Once the cells get to a given size, we want them to divide. And that's a little bit more complicated, although not too bad. We have to create a place where we're going to keep track of which cells divide at each time. That's called cells to divide. We loop over all of our cells again, as usual, for cell in self.celllist. If the volume of the cell, now this is the actual volume, not the target volume of the cell, is bigger than 50, we say that we're going to have the cell divide. So we add that cell to the list. Once we've made a list of all the cells that are going to divide, we apply a cell division operator to each cell that's going to divide and have it divide. Once the cell divides, it has to decide what to do, because we started with 1 cell, now we have 2, and we have a function called update attributes that tells how space is divided between the parent and the daughter cell, and what attributes the daughter cell has that are the same or different from its parent. And so here we have a function, update attributes. We assign half of the volume of the parent to the child, so the 2 daughter cells have equal sizes. We say that all of the properties of the child cell are the same as the parent. We clone the parent onto the child, except that we have this little feature here that we say that if the parent cell was of type 1, was cell 1, the daughter is of type 2. If the parent was of type 2, the daughter is of type 1. And that's the full specification of the simulation that we just ran. And again, that was just a quick fly through. We'll come back and spend a week teaching you how to build more serious simulations using that. But that's about the level of complexity that we're going to be dealing with. It's not very complicated. It's not trivial by any means. It takes learning, but it's not very complicated. Almost nobody comes to this kind of class knowing everything that yes, we'll talk about parallelization. But literally, to parallelize your code, you take the line where it says number processors and change the number. That's all you have to do. It's intrinsically parallel. We can talk more, we'll definitely talk more about that. Nobody comes to this class knowing, usually, unless they've done multi-cell simulations before, nobody knows all of the things that you need to know to build multi-cell models. And so one of the pleasures of this class is that some people come knowing a lot of biology, some people know a lot of computer science, some people know mathematics. You don't need to know a lot of detail about any one thing, but you do need to know something about a lot of different things. And so, for example, if you're thinking about biology, it's really important to think a little bit about what cells are and what they do. In particular, how do they apply forces? How do they move? And how do they build tissues? So we'd like to have some basic understanding of cell biology and tissue biology. There's some key concepts like mesenchymal and epithelial tissue, cell polarity, planar polarity, extracellular matrix. If those are unfamiliar terms to you, it might be worth reviewing a little bit of biology, basic biology before the class. If you aren't familiar with signaling, metabolic, and regulatory networks and how they're drawn and how they work, it might be worth doing a little bit of review of that. Herbert Sauro is going to be teaching a course on that next week. I know some people who are going to be taking our course are taking his course. Over the years, we typically taught those back to back in consecutive weeks. This week, they're going to be separated by week. You don't need a lot of knowledge about these things. Think of a Wikipedia level knowledge. In mathematics, if we write a summation or an integral, it would be helpful to be able to be comfortable with that. We're going to be doing a certain amount of first order differential equations, rate equations, nothing very complicated, but basics. How we represent chemical reactions and regulation is important. The only partial differential equation we'll use is the diffusion equation. And so familiarizing yourself with the diffusion equation wouldn't be a bad idea. We'll have some dynamical systems concepts of stability and fixed points. We may do a little bit of sensitivity analysis, and these are stochastic models. And so understanding stochastic rates is helpful. Again, you don't have to have had a whole class on these subjects, just a basic passing familiarity. In class, we'll cover them as we use them, but because the class happens quickly, it's helpful to have seen them once before. In biophysics, I've already talked about the idea of a constraint and an ideal gas and a spring. And so thinking about constraints and how they work is important. What are called constitutive relations, that is how materials behave, and a spring is an example of that, are useful. If you're interested in understanding the core computational methodology, cellular POTS or GGH model, that requires a little bit of a knowledge of statistical physics. You don't have to have that in order to be able to use CompuCell or to get it to work for you. But if you're interested, it's certainly helpful to have. Most of the Python that you need, we've already covered. We've covered how to do conditionals. We've covered how to do loops. We will talk about the two object classes that we use all the time, which are lists and dictionaries, which may be less familiar. And one of the things that you may run into in Computacell a lot is self-dot. Why is there a self-dot? That's an issue of what's called scoping, and we'll talk a little bit about scopes as well. If you haven't built models before, thinking about how you conceptualize models. How do you take a biological problem and turn it into mathematics or computing that can give you a meaningful answer is not trivial. And so thinking about what it is you want to model, what it is you want to predict, what's a meaningful model, these are things that take effort, and I'd say are probably the hardest part of building biological models. And so you might want to think about, from the point of view of the things you want to do, how to answer those kinds of questions. If you want to fill in background, we're going to be teaching that Python bootcamp. There's this great online Python course called Rosalind that I recommend. It's free. It's really good. There's a good introductory Python textbook, but there's a link here. For network modeling, the online documentation for Tellurium gets you started really well. Herbert Sauro has a great course called Systems Biology Introduction to Pathway Modeling. Great book. If you want to do more dynamical systems and mathematical biology, Nick Britton's book, Essential Mathematical Biology, is a great compact introduction. On biology itself, my go-to place is an old edition of Gilbert Developmental Biology. The new editions are very expensive and they're very long. If you buy a 20-year-old edition, it's much shorter. It has all the things you need in it. It costs $2 online on Amazon. So I strongly recommend going and buying a 20-year-old copy of Gilbert. It's really a helpful thing to have. For biophysics, there's a good introductory textbook by Phil Nelson, Physical Models of Living Systems. If you want to grapple with the complexity of real biophysics, which is hard but beautiful, the best textbook by far is Physical Biology of the Cell by Phillips. Beautiful, beautiful book, but quite demanding. On model conceptualization, there's a nice book by Gerli and Lund called Scientific Models. That's very elementary, and it's not focused on biology, but it does talk about how model conceptualization works. It's quite nicely done. There's that 10 Simple Rules text paper that I recommended. And there also is a document that I put in on how to build models that's in the readings. It's not published. So if people come up with any sources or suggestions for websites, books, articles that they think are useful introductions to any of these topics that you liked and that were meaningful to you, please let us know, and we'll do our best to add them to the repository so that other people can benefit from them as well. We're always looking for new ways to cover this material. In terms of preparing for the class and the hackathon, I hope everybody can get CC3D running on NanoHub. If you want to run it locally, that's okay, but you don't have to. As I say, everything we do, we can do on NanoHub. You can try loading, editing, and saving some CC3D simulations. You could start with the one that we just created. You could try changing some of the parameters in that model and seeing what happens. You can load new models. If I want to load a model, I go to Open CC3D Project here. And if I look in my directory, I will find somewhere something called Computacell CC3D demos, and it will give me a list of folders which are rather mysteriously named. Our architecture here isn't very good for navigation, but I could go here, for example, to one of these demos, and I can run that demo. If you're looking in the demos, steppable-based mitosis is a nice one. Angiogenesis for blood vessel development is a very nice one, and delta notch is another nice one. The nice thing is you can't break anything. The worst thing that can happen is that the system crashes. You have to hit terminate and launch Computacell again. So I encourage you to explore. There was a question about how to change color schemes in Player. You can try the configuration tools here and see what happens. General settings, colors, how to save movies. We'll go over all of those things in the class, but if you played with them a bit, it certainly will make it easier when you go over it again in class. And background reading, I've talked about some of the things that you might want to have a familiarity with. As I say, almost nobody comes to a class like this with familiarity with all of the things we're going to work with. And so if there are any of those topics that I covered very briefly that seem unfamiliar, you could look at some of the resources, or you could do some Wikipediaing, or you could find new material that you think are useful. And the last thing is, if you want to do the hackathon, I hope people will. You don't have to sign up for it in advance. If you signed up for the regular class and forgot to sign up for the hackathon, change your mind, you want to do it, that's fine. Do think about a project you might want to do, or what kind of project you might want to join, and post your ideas on the Slack, Google Doc. And if you're going to propose a project, please do send us a single PowerPoint slide about it. Okay, so that took a little more time than I promised. It's a little over an hour. But if there are any things that people want to discuss before we break, I'm happy to, Jim and I are happy to answer questions now. So I hope that was a useful fly through of some of the things that we're going to address over the course of that week. Okay, are there anybody have any questions or comments before we stop? No?
Can I ask a question?
Please, go ahead.
Is it okay if I talk into my microphone? I can type it in if you don't want me to.
No, you can talk.
All right.
The only time if we have 150 people in the room, then muting the mics is necessary. But in a small class like this, there's no reason we can't just talk. Please go ahead.
Makes sense. So first of all, wonderful introduction. Thank you so much. I just have a very general question. It seems like this software is addressing the interactions within a cellular scale. Is it like when you talk about mechanics and like interactions, like you're not talking about, like, for example, the mechanical behavior of the tissue as a macro scale object? You're talking about the mechanics between cells. Am I right?
Well, you can certainly, if you wanted to build a tissue model of a whole limb, for example, or a whole brain, then you probably wouldn't divide it into single cells because there'd be too many of them.
Yeah.
So for simulations of that kind, people typically use continuum mechanics models. There's a nice package, for example, called FE Bio out of the University of Utah that's very good for that.
Yeah, I personally use Phoenix for that, but I wanted to see if this program has the ability to integrate to other, like other softwares or something like that. Is that possible?
So the use of links allows you to build a finite element like solids models inside of CompuCell. So you can use your cells to represent blobs or volumes of material with compressibilities. And so you can build solids models using CompuCell. It's not the most efficient way to do it. A solver like Phoenix will do it better. But you can actually solve fluids and solids models inside of CompuCell if you want to. The integration with Phoenix is interesting. We tried, but Phoenix for a long time would only run on Linux machines.
Yes.
And so that ruled it out for us. Maciek actually spent several years trying to convince the Phoenix community that they should acknowledge that Windows existed, and there was a lot of resistance to that. He did do a Phoenix CompuCell integration, but because we couldn't release it to all of our users, and the majority of our users I'd say 95% of our users either use Macs or Windows. it wasn't something that we could support. But I'd love to see an integration with Phoenix. And we've also talked about trying an integration with FE Bio. There have been teams that have done integrations of CompuCell with other solvers of a variety of kinds. People have integrated CompuCell with some radiation biology solvers to do cancer treatment simulations, for example. And so if you were interested in discussing that, I'm sure TJ would love to talk to you about it. That kind of integration is probably outside of what we can do in the class here. But it's certainly something where if you see ways forward with that, that would be great. Yes, a containerized version of CC3D in Phoenix would be very interesting. If you could put it in Docker and then run it, that would be great. I'd encourage you to talk to Maciek and to TJ about that possibility. It's certainly something that we've wanted to do for many years.
Yeah. Yeah, I agree with you. Phoenix is not very, ubiquitous, as like it's just at some point it was just on Linux, and then they offered the Docker, and then it's just a pain to actually have it on a Windows computer. So yeah, I understand, but yeah, a container version might be a compromise here. So I don't know about that. I've never seen something like that. Like Phoenix usually doesn't integrate with other things like very well. Might be the reason.
Maciek did have it connected, but again, it's quite a while ago, so we'd have to go back to talk about that. I mean, it's several years. It's several years ago that we did that. Somebody says they're looking for the Gilbert Developmental Biology. As I say, I do recommend that. It's a great textbook. Beautiful, beautiful textbook. And as I say, you don't have to buy the latest edition, because the latest edition of Gilbert will have all sorts of molecular species names in it, which you don't care about for modeling. The basic things cells do about dividing, differentiating, dying, signaling to each other, those things have been known for a long time. And so Gilbert from 20 years ago for $2 works just fine. And so it's great to see that some people are coming from a very computing background, some people come from a very biological background. And I hope that during the course, even though we're separated and even though it's going to be by Zoom, that people can find people that they can share ideas with, and that people can partner a bit so that somebody who knows a lot of biology can help somebody who has a strong computer background and vice versa. When we did this in person, we'd always try to team people up. We would teach this in a beautiful conference center with big tables, and we'd say, okay, you're a biologist and you're a computer person, so sit next to each other and help each other out. It's a little harder to do that by computer, by Zoom, but I hope at least during the hackathon part of the course we can do that. I do welcome the fact that we have people with clearly very strong computational and solids mechanics background, people with strong biological background. That diversity is the strength of the CompuCell community. It may mean that sometimes we go a little bit slower in our presentations or don't cover as much detail as people would like. But remember that everyone is learning. As I say, very few people, there are few always, but very few people come to this course knowing all of the material that they need. If they did, I suppose they wouldn't have to take the course. And so I hope that we can learn from each other, and that it'll be a fun week for everybody. Any other questions or comments before we break? It's getting pretty late here, 9:30. People are troopers to be here. We gave this presentation at 8 o'clock this morning for our European colleagues. It was interesting that for the first time we had a number of people who signed in by Zoom from Africa, which I was delighted to see. We had someone from South Africa, I think somebody from Nigeria signed on. So one of the nice things about Zoom rather than doing it in person is that people who wouldn't be able to come otherwise are able to participate. And so there always are trade-offs. Anything else before we break? I hope that was helpful to people. And I am looking forward to working with you a lot during that week. It's pretty intense. And I hope that at least for some of you, it winds up being something that you can use going forward in your careers. I guess then I'll say good night to you all. And look, thank you again. I'm looking forward to working with you.